Well, thank you, Corinne, and thank all of you who are willing to stay on here for the bitter end. So I feel a big state of responsibility here and almost an overwhelming expectation of how I could bring to bear a kind of a summary of the extraordinary information that we've been privileged to be exposed to over the last few days. It's really been quite a remarkable compressed neuronal stimulation for everyone in this field. And so with homage and with a caveat, I probably need to give a disclaimer to begin with to say I'm not going to do an adequate job probably of really fully describing the material that you've been exposed to over the last few days. But I'll try to give a go at it here with the construct of Back to the Future. So this is a little bit of a Back to the Future, which I feel that probably is the one unique feature that I can provide because I've been involved in this field since the 70s, since the middle 70s. So I do have some perspective on time that looks back to look forward. So I want to start first with just thinking about the concept of toxicity from a very broad-based perspective, just to set the context. So if you think of this cellular soup, Rob and I talked about the saline versus the non-saline environment and where we came out of the oceans as unicellular organisms and then into polycellular organisms, and then we started to be able to, I'm running through billions of years of history here, geological history, but then we eventually started to climb out of the water onto the cooling land masses, and eventually this toxic gas, oxygen, that started to envelop the environment as the green plant period started in full swing, then gave rise to the ability of an organism to start using oxygen successfully to its advantage. And it gave rise to air-breathing organisms, aerobic organisms, and all the metabolism that we study. Throughout the course of that developmental history, and again, I'm taking broad swaths here of history quickly, was the commensalization or the intercommunication between the microbial world and the polycellular world, this eukaryotic and prokaryotic world mixed. And so the organisms that were growing up as eukaryotes in polycellular form were carrying along with them these prokaryotes. And one interesting example of that, of course, is most of you are familiar, the emergence of the mitochondrion. The mitochondrion, we feel, was an endosymbiotic organism that was a probiotic that have found a friendly place to live inside a eukaryote and was able to then produce energy for the sake of the eukaryote, but also had a warm, friendly place to live. So we had this interesting interrelationship at the cellular level between prokaryotes and eukaryotes. But then we also recognized that there were whole organisms of prokaryotes that were living inside or living on or around the eukaryotes. And so this symbiosis or this commensal or even this parasitic relationship can develop. So those are three ways, right, that the prokaryotes could live in community with the eukaryotes. It could be either symbiotic, that's like reinforcing positive nature of the relationship. It could be commensal, which is kind of neutral, maybe not positive, but not negative. And then there was this parasitic relationship. Now, what is this parasitic relationship all about? Now, again, I apologize. I'm taking wide sweeping assumptions here, but I hope it'll make my point as I get through it quickly. The parasitic organisms are parasitic because obviously they are doing something as a detriment to the host organism in this case. That's why we call them a parasite. So what kind of things could they do? Well, they have their own genes, and they have their own metabolic uniqueness, and they therefore produce their own secondary byproducts of their metabolism, which are their own intermediary materials that make them up. Like if they're prokaryotes, they have cell walls. We don't have cell walls in eukaryotes. We have cell membranes, so there's a difference there. And those cell walls are composed of a very interesting class of mucopolysaccharides, lipoproteins that we call inflammatory agents in the human cell, right? So we have these interesting cell wall components in certain prokaryotes that when those organisms die and they release their cell wall debris into the environment, that those have been found to be toxic to the host if it's a eukaryote. And so these lipopolysaccharides, LPS as you've heard them, are of a whole different family of different molecules that relate to the cell wall structures of gram-negative and gram-positive bacteria that have different relationships to the host organism, in this case, the eukaryote. Now, what that leads then, and here this is the payoff for having to endure this introduction, is it leads to the fact that now we have an organism living there that is producing a byproduct or a part of its metabolism that is not so healthy for the host, right? So I will call that, for the sake of discussion, a toxin, right? So now the organism that is playing host to that prokaryote has to find, over time, if it's going to be selected for living in this environment with oxygen that's hostile and all these various things that are going on, it has to find a way to get rid of those secondary metabolites that are producing harm. So that is a process called detoxification. So the evolution of the cytochrome P450s is a very interesting chapter in the history of evolution. If you think about it, it goes back long, hundreds of millions of years. And those cytochrome P450s are unique in their ability to use oxygen as a defense against the toxins that they're being exposed, right? So now, why do I know that? Because these cytochrome P450s are iron-containing enzymes or proteins, right? And the reason that we know that is P450 stands for the spectroscopic lambda max of absorption in light of those enzymes. That's how it was named, cytochrome P450s. 450 is in the visible range of the spectrum, and you know blood is red, iron is, and it leads to those chromophores, right? So the chromophore iron, or the metallo ion, in cytochrome P450s gives rise to their color. Now, this is very interesting. I could go on and do a riff for some time. I'll beg that off. But it's interesting if you think about, in studies recently in beiging of fat or browning of fat, just to show you how this works. So if you take an animal, a rodent is the best example, and let's say we take a nude rodent, no fur, and because there are species that have no fur, and you then put that animal in the cold. You subject it to, like we're seeing a lot of people do now with ice bathing. What you will find is, if you look at those animals visibly, without going into high technology, you will find that their skin starts to get brown. It starts to get dark color. And the reason for it is that the subcutaneous fat gets beiged because it activates gene expression of iron-containing cytochromes that are involved with a fetal reaction that produces energy from heat by uncoupling mitochondrial oxidative phosphorylation. Okay, so that's just a visible example of how an organism can adapt its detoxification machinery to a changing environment. Now, I'm using the term detoxification a little bit broadly there because these are really uncoupling proteins of mitochondrial function, but they're the cytochromes that are associated with this whole family that we talk about with detox. Okay, now, why am I taking you on this voyage or this journey? Because if we then think, as you have seen over the course of the extraordinary presentations of speakers over the last few days, of what toxicity means in the biggest generic sense, it means endotoxicity and exotoxicity. I mean, those enzymes that are involved with detoxification didn't know they were going to be exposed to the whole array of petrochemical and polynuclear hydrocarbons that we've produced in our chemical factories over the last 100 years. But what they did know is that there was going to be all these various chemicals that would require specific types of deconstruction machinery to break them down, a lot of which came from endobiosis, from the endotoxins that are produced by microbial metabolism, as was shown by Rob Knight when you start doing metabolomics associated with genomics of the microbiota. So when I think of toxicity, clearly, in the broadest capital T, I'm thinking of the combination of endotoxicity and exotoxicity. It's that accumulation, we sometimes call that the total load effect, right? And so a patient or a person who experiences some kind of a toxic-related effect is in the detective work to figure out how and why, you have to look at the endotoxic load and you have to look at the exotoxic load because they're being funneled through similar pathways of resistance or resilience, these detoxification pathways. And as has been pointed out by multiple presenters over the last few days, these are highly polymorphic processes that have unique genetic underpinnings. So we don't have the same response to the same material for all people. Okay, with that then said, then you start to say, well, gee whiz, now you've kind of broadened the pie, or you've made this bigger in terms of exo and endotoxins. So how does that interrelate with these whole range of environmental considerations that we're dealing with and the clinical presentations that we see, and how does that ultimately lead itself into personalized nutrition? So yesterday, or the day before, I think it was the day before, I made a wild statement, which I've gotten to think a little bit more now, that maybe I wasn't as wild in my statement as I thought at the moment, about these barrier disorders that we're experiencing clinically now that are becoming frequent challenges for the old model of one thing against one disease for one pill to treat it. And so what are those rising tides? And I gave you a list yesterday. I went back to check myself to see if I was right, and I think I was. These are conditions that over the last, say, 20 years have been rising in prevalence dramatically while our genes have remained the same. I want to emphasize we haven't undergone huge genetic changes, maybe epigenetic changes, but not fundamental sequence changes over those last 20 years. So what are those? This is kind of a partial list, and that list you know of these. So it's Alzheimer's, autism, non-alcoholic fatty liver disease and its companion non-alcoholic steatohepatitis, thyroiditis, idiopathic pulmonary fibrosis. Idiopathic means we don't know the origin, but yet it's rising about 5-fold over the last 10 years. Inflammatory bowel disease, and the most common reason that people present to doctors, sinusitis, which is exponentially growing in prevalence. Now, what do those conditions reflect in the light of what I've just talked about with endo and exotoxicity? Could those be conditions that are associated with the way the inside and outside environments are interacting with receptor sites that create alarm messages for which the body then does what it's supposed to do, right? It's not like a disease. It's related to the function of the body trying to protect itself. And some people have more genetically sensitive protection mechanisms than others. Now, we have historically maybe said those are people with genetic imperfections. I would say they're not genetic imperfections, they're genetic uniqueness in the way they're responding to their own burden or their own response to the load of these various substances that the body's designed to try to get rid of. So I think we do a little bit of a disservice when we start labeling genes good and bad, and we start saying, well, you know, that person has the bad genes for detox and that person has the good genes for detox. They have the genes of uniqueness to their own environment that are built on the legacy of their millions of years of history going back to, you know, the dawn of civilization. And so it's not good or bad, it's uniqueness that then leads to personalization. Are we all? So I think that what's happened in our field, unfortunately, in genomics, is that we start labeling things as good or bad. Let me use another example. I know I could go way off on this, but just bear with me for a sec. And that's ApoE4. As we know that there is this very strong clinical correlation between a double ApoE4 and both atherosclerosis and dementia. And so we are so concerned about that because the risk profile of a double E4 seems to be so associated with the phenotypic presentations. This is an phenotype into these diseases that people often don't even want to know that on their genetic history because it's been given this horrible reputation of being like a death sentence to dementia. And actually, that's not true. If you really study ApoE4 closely, what you will find is that ApoE4 was probably the predominant genotype of the ApoE allele back in early history because it is a pro-inflammatory state that gives that person a quick response to infection, which is probably one of our biggest threats to early civilization. We didn't have antibiotics and medicine and all the stuff we have. And so organisms, if they couldn't survive against inflammatory injuries or infection, they didn't make it to the reproductive years. So ApoE is a heightened response element to inflammatory modulation. Now, in some cases, maybe that allowed that person with an ApoE4, their ancestor, to survive, that they wouldn't even be around without carrying that gene. And so what do you do then if you have a gene that is a heightened sense of inflammation? It means you better be really concerned about things that would turn on inflammation. You have a heightened need to personalize your program to stay away from precipitating triggers that would initiate ApoE4 becoming disadvantageous for you rather than serving a useful purpose as to protect you from infection or inflammatory processes. The point I'm trying to make is that I think we need to distance ourselves from the value equation of good and bad and move into the uniqueness of susceptibility or unique response, and define personalization as a value-free term relates to finding the right environment for the right person to get the right outcome. Am I making sense here? Okay, so with that as a precept, by the way, you don't have to agree with anything I'm saying here. I get the privilege of just spouting on, but I think there is some value in at least considering this model, as I'll go on to talk about. So how does that then relate to clinical nutrition? And I'm just going to give you a couple of slides, and then I'm going to go back to the future, if you'll bear with me. So the subtitle of this, you'll notice, is It's All Changing. And the reason I say it's all changing is that we're still teaching out of textbooks that are so artifactually wrong that it's almost embarrassing to be thinking that we're teaching young minds this old stuff. And maybe even embarrassing is too light a word. There is so much that has changed, as you can see, just over the course of this last few days. And I mean, all one needs to do is listen to Rob Knight for about 5 minutes to recognize the rate of change, right? It's just exponentially changing. So to think that we can teach out of textbooks around what nutrition is that are built around premises of 2000 is ludicrous. But that is what's happening, and we're turning out people that are going to guard the gates of this knowledge because they had to study for tests, and gee, well, they don't want to think it was all for a loss. And not everything you're learning is wrong. I mean, there's fundamental things that we recognize are true today that were true 50 years ago, but there's a lot that's not. So I'm really pretty excited to see that this personalized nutrition, this concept that many of us have been advancing as a thought for many decades, is starting to get some traction now. In fact, I went back as I was sitting there in the audience and thinking about my first really extraordinary transition into this field when I took a sabbatical in 81 and 82 with Linus Pauling and the Pauling Institute, in which my responsibility as the director of one of the research labs was developing gas chromatographic mass spectrometry interface systems to measure urinary metabolites to try to do metabolic patterning for orthomolecular psychiatry. That was what was my project for 2 years at the Pauling Institute in Palo Alto. And there were, I could go back and tell you about all the issues that we had, of both technical issues of developing the machinery and getting precision and so forth, and then the variability that we learned among individuals that made that kind of research go from group population-based to think maybe we need to do N of 1 studies of each individual against themselves over time. So that's a whole chapter for me that really, when I left, actually some of you know, I decided to give up my tenured faculty position and to go out into the world to try to teach physicians about the future of what personalization was going to be. That was a pretty bold step. I gave up tenure. My parents thought I was crazy. I had a family. And here was my advocate saying, no, we're going to do something else. We're going to teach people how this new technology can be applied clinically to improve patient outcomes. Well, over the years, now we see a whole bunch of companies. So there's an industry emerging. And I would say to you that you can never have something that is going to catch and stick if you don't have a commercial engine behind it. And I would say that great ideas are powered by capital, and capital is potential energy. Energy coming from capital is agnostic to its value. It can either be used for good or bad. And I would like to say that the energy that underlies this industry can be used for good to create the value proposition of this transition. So in the absence of an industry, you have no ability to stick these things and grow them up. But now we're starting to see an industry to develop. And it's been speculated that over the next 10 years, this industry of personalized nutrition will be a several tens of billions of dollars industry. Several tens of billions of dollars industry. In fact, I will go on, and you can hold me accountable to say that hopefully, even within my own lifetime, if I'm so lucky, we will see this technology rise up and be so well adopted that it will spawn an industry that will be like the microcomputer industry does in the age of the garage stuff of Wozniak and Jobs and Gates and Paul Allen. That who wanted computing? Who wanted personal computing in 1975? It was only we hobbyists, we weirdos, that went down and got those Alltech computers that we could build at the hobby store and start playing around. I can recall my my middle son working on a project with me for his 8th grade when he was at Palo Alto in school. And we were writing code on a little floppy disk thing that was supposed going to analyze fish behavior. And so, it's really, the revolution that's occurred is that people don't think they're computing, they're solving problems that they have need for, which is what we're going to have with personalized healthcare. It's going to solve a need. People won't have to know all the its and ups and the ups and downs and the nitty gritty. They just need to know what it delivers as a value proposition. So I happen to, one of the places I always look to where the frontier of these things are changing is in the veterinary area, because the vet scientists are a lot more advanced in nutrition than are the human nutrition scientists. And there's many reasons for that, because they can do the studies more easily, they can hold them under constrained conditions and so forth. But I don't know if you've been following the largest pet food company in the United States that actually has been, as you probably know, acquired by Nestle a number of years ago. That's Purina. Purina, the number of vet scientists they have on their facility in St. Louis is equivalent to all vet sciences of all other industries in the pet food combined. So it's a very, very big, huge facility. And just to show you what's going on that I think is interesting at the leading edge, you probably have seen over the last couple of years 2 products coming out for animals, one of which is this calming care product, which is a product for particularly barking dogs, because barking dogs is a measurement of stress, a neuronal stress of the dog. By the way, I didn't know any of this until I started getting into the literature. And so if you want to reduce stress on a dog by neuronal stress, by the phenotype of barking, to reduce barking, then you want to somehow calm neural inflammation, right? And they have a product for that. And would you know that it's a probiotic product? You're probably familiar, this is the product. If you've not studied it, just go to their website and take a look at this brain-gut connection that is in this product. And then they have this other product for aging dogs for cognition, right? And if you look at these are all research, and they have published studies, and it's highly, what they didn't just come out of the blue air, they really are studying this. And this Bright Mind product has a list of ingredients that you would think might be in a Dale Bredesen protocol, right? So the point I'm trying to say, you have to look out there and ask, where are the changes occurring, and how do they get transmitted and communicated into population at large? So why is this change in thinking happening so rapidly? And we only need to look at the September 24th issue of JAMA to kind of see the nature of change. This was an editorial that was published called U.S. Dietary Guidance. Is it working? And this is kind of a rhetorical question, and the answer is no. Okay, and the reason that it's not working according to this, I'm summarizing here, so please apologize the summary, but the reason it's not working is that we've been using community-based population studies for years to try to describe how individuals respond to their diet. And first of all, that messaging, those public health messages that are based on population studies, are not compelling enough for people to really make a change. So the frequency of behavior change is very, very low, because it doesn't address the individual needs of the person adequately to feel their motivation will stick to something. Secondly, the evidence that are being used for these guidelines are built around the precepts that we can take epidemiology, which is a population-based information system, and we can convert it into understanding an individual. And we all know that that actually is not true. You cannot, all you can develop is hypotheses from epidemiology. You cannot generate specific action points as to its application. So we have a whole system built around association and not causation. And in causation, we have to go to the individual. So that's what this article says. With continued investment in precision nutrition, future dietary guidelines might incorporate an approach that's built around optimizing biology, perhaps extending age-specific guidelines to other subgroups defined by certain biological factors. Do you get what I'm saying there? Certain biological factors mean you need different information. You have to ask different questions to get different answers in the personalized nutrition space. Okay, so what that's all built on is this extraordinary revolution that, again, we've had a number of very good presenters that have talked about this over the last few days, about this gene-nutrient phenotype connection. This is a dramatic change in our thinking that's come out post the Human Genome Sequencing Project.
So here's how I summarize this. This is a Jeff Bland summary, so this is not exactly what they say in this issue of Science, but they imply it, I believe. We are a food-based culture, right? And that's a shared common human experience. I've never met someone that doesn't eat sometime. Just as I've never met someone that doesn't breathe sometime. And I never met someone that doesn't consume water sometime. So there are certain shared common experiences that are essential for function. So if we're a food-based culture, physiology is determined by the nutrients we consume, whose need for those nutrients is determined by the genotype of the individual. Therefore, my ipso facto is, food represents the delivery system for nutrients that modulate an individual's phenotype, which is what we call nutrigenomics. So that's how it all travels down the thinking for me as it relates to personalization. And in this article, they have this, or in this issue of Science, they've got this really interesting graph under the title Translational Genomics and Precision Medicine, Moving from the Lab to the Clinic. And in this, they talk about this one sector, which they call prognosis, with biomarkers. Now, I often hear in our field people use the term diagnosis. They use a certain kind of set of tests to diagnose nutritional inadequacy. We're not using it to diagnose anything, I think, unless you're trying to diagnose scurvy or something. But what we're using them is to prognose a forecast of the trajectory of that person's physiology going forward. And we're using these information systems, genomics, omics, biometrics, and informatics. We're using those systems to prognose the trajectory the patient is going toward, so we can then help modify if that projection is going in a direction that we don't think is in the best outcome of their health objectives. So prognosis is really what we do, and it's not nutritional deficiencies, it's nutritional insufficiencies, as Roger Williams talked about with genetotrophic diseases. It's not that we're all walking around with beriberi, scurvy, pellagra, xerothalmy, and rickets. We're walking around with subclinical undernutrition, overconsumptive undernutrition that modulates our function in such a way that our physical, cognitive, emotional, behavioral, and metabolic function is adversely affected. That is the Institute for Functional Medicine, by the way, what I just said in a summary. Okay, so with that, then we had the fortune at our recent meeting to have Albert Laszlo Barabasi come and talk to us. And so Barabasi, author of this book last year called Network Medicine, Complex Systems in Human Disease and Perspectives, and Therapeutics. I was very flattered, quite honestly, because Dr. Barabasi spoke after I kind of gave my introduction to the concept of our meeting, our personalized lifestyle medicine meeting. He spoke next and did this absolutely brilliant presentation. And then later, he came up to me and says, "Jeff, I got to have your slides." And I said, "Why?" He says, "That was the most complete discussion and information I have ever heard that interfaces network biology to nutrition. And I want those slides because I want to incorporate this information in this new $100 million research grant that we have gotten that I'm going to be working on at Harvard and our collaborators over the next few years." So I think that there is stickiness to this model once we can describe it accurately and in a way that people understand. Now, there is another on this slide. There's a first page of an article. And the reason I put that first page of the article is this is a recent co-authored paper from Barabasi and others. And let me tell you how this affects me and how help affects you, because this is a segue now into Back to the Future. And I have, 25 minutes. So the title of this paper is Uremic Toxin, Indoxyl sulfate, promotes pro-inflammatory macrophage activation via the interplay of this OATP-B21 and Notch signaling genetic network system. Okay, now, most of you probably would maybe see this article and say, "Oh, let's turn the page." That doesn't sound very interesting. Let me tell why this is Back to the Future for me, because this is Eli Metchnikoff revisited. Now, let me just take a breath here. So what happened to me in 1977, as a professor at the University of Puget Sound in Tacoma, Washington, I was asked by a group of renegade young naturopaths who knocked on my door, the leader of who was Joe Pizzorno, and in tow was Dr. John Bastier, who then asked me if I would teach
a course to them so they could be relicensed in the state of Washington. Without that course taught at a university, they would lose their licensure in the state of Washington as naturopaths on herbal pharmacology. Now, this is ludicrous for me to say that I actually undertook to do that when I knew virtually nothing about herbal pharmacology, but I can tell you I learned a lot from those students over the course of the next 6 months that they were in this class, because I worked really hard to kind of come up to speed. But one of the things that I learned in that course was about Elie Metchnikoff. I was so excited about learning about his concepts that I ultimately now have in my library an original signed copy of Elie Metchnikoff of his book, Prolongation of Life, that was translated from the French into English in 1902. He won the Nobel Prize, you know, in medicine in 1906. And how did he, and why did he win the Nobel Prize? He was, by the way, took over the directorship of the Pasteur Institute after Louis Pasteur had passed on. And what he is credited as discovering, and there's some controversy as to whether he was the first person or but the second, but anyway, he was certainly worthy of a Nobel Prize for his discovery of innate immunity, the innate immune system. And how did he make that discovery? Presumably, this is a story that's written. He was at the French seashore, and he was doing beachcombing, and there was a starfish there, and there was sea urchins. And he took the sea urchin, and he punctured the skin of the starfish, the under part of the starfish that's a little easier punctured with the quill of the sea urchin, and then he had his monocle, and he watched, and he saw these corpuscles aggregating at the site of the puncture. That, remember, he came from the age of bacteriology, so he's thinking, well, gee, are these bacteria that are, no, they're way too big for bacteria. I can see them with a magnifying glass. I don't need a microscope. So then he wondered what the heck they were. Well, obviously, macrophages later. So he started to really study then the origin of the innate immune system. Now, the reason that we now are so interested in the innate immune system is that much of what is going on in our gut microbiome entails the communication between these gut bacteria and the gastrointestinal-associated lymphoid tissue and the mucosa-associated lymphoid tissue, the galt and the malt, where we have all these different species of our immune system waiting with open arms to pick up information from our gut, including all the debris that bacteria are producing, as you saw from Rob Knight's presentation as it relates to the metabolomics of what's going on at the gut microbiome level. So the immune system is being modulated in part by all these things that are happening in the gut. We all know this now, but I can assure you in 1906 that was not known. And he wrote this book, Prolongation of Life, and probably most of you know what that book advocated was the use of instillation of Lactobacillus bulgaricus by enema for the treatment of age-related diseases, right? That was what that book was about, prolongation of life. And in this construct, now I'm going to roll forward. You knew I was going to come back to this article, so now we're coming back to the article. So back to the article, one of the chemicals, one of the toxins that's produced by going to our previous presentation to a tryptophan metabolite is a material called indoxil. Indoxil has, in the ancient language of natural medicine, naturopathic medicine, was called indican. They didn't actually know the chemical structure at the time that it was given that name, but what they knew was in the urine of people with endotoxemia, even before it had the term endotoxemia associated with it, that you could, by putting hydrochloric acid and phenolphthalein in there, you would create this blue color in people that had endotoxemia. This was the classic indican test. The indican test is a simple colorimetric test for the production of indoxyl sulfate. Now, where did the sulfate come from? That is a detoxified product of indoxyl by the sulfation with phase 2 detoxification enzymes. Now, what does this paper say? And by the way, this was very cute. When I talked to, I think actually Dr. Barabasi said this on the podium when he walked up. He said, I'm so glad that Dr. Bland put that paper up because I really didn't know exactly what I was researching. He didn't realize the history of this indoxyl sulfate, which is a uremic toxin that is related to macrophage activation and kidney inflammation, right? So now we're kind of building an interesting story around what is toxicity, how does it relate to function, and we built in now the concepts of detoxification. Okay, now, if you'll hang with me for a moment. Now I'm going off the slides, now I'm going on a riff. So hang with me, this is a riff.
So one of the things that we know that I think is really interesting that relates to these disorders that I talked about that I call barrier function disorders like Alzheimer's and autism. Now, why would I call Alzheimer's a barrier function? Because the brain is interconnected to the rest of the body through the barrier called the blood-brain barrier, which we now know if there's leaky gut, there can be leaky brain. So we start looking at membranous compartmentalization of the body and how that is differentiated in a state of inflammation and in a state of toxicity. And so we start to think, well, hold up, maybe things that were originally prohibited from being exposed to the outside world now become exposed to the outside world through these processes of becoming leaky, becoming permeable. And so when you start looking at all of these disorders, remember the brain is the one place in the body that is the one place in the body that's directly related to contact with the brain is the nasopharynx. Those horns of those nerves go directly to the brain. So you think of all these respiratory disorders, but you also think of brain-related inhalant toxicity, which we often been talking about ingested toxicity, but inhalant toxicity, particulates of small particle size can deliver directly into the nervous system these loads of materials that need to be, quote, detoxified. So with that as a history then, I'm sitting in my office. This would have been 1982, when I was just coming back from the Pauling Institute and going to be starting into this new adventure. And a friend of mine said, Jeff, are you familiar with Armand Quick's work? And I thought, no, I don't know Armand Quick. And he says, well, he's deceased now, but Armand Quick, who has been given the credit as being kind of the person who first, in New York City as a pathologist, the first person in the early 1900s that really discovered hemorrhagic disorders and coagulation disorders. He's very, very famous. And if you Wikipedia Armand Quick, you'll see this. But he also did another interesting thing that you might be interested in, because I was talking about endotoxemia and Elie Metchnikoff. He said he actually was the guy that made a discovery of patients in New York City hospitals that had acute liver failure, that you could assess their liver failure by looking in their urine for various types of metabolites that were related to glycine conjugation, specifically hippuric acid. Now, what is hippuric acid? Hippuric acid is a chemical that is a combination of a breakdown product of benzoic acid and the attachment of the amino acid glycine in phase 2. So it's a kind of a measurement of a phase 1, phase 2 outcome. So I said, really, is that true? He said, yeah, go look in the literature. I challenge you, go look at Armand Quick's literature in 1936. You can still find it on PubMed. And he found out, and there's actually one paper, it's really interesting. He's got 27 different patients that are all hospitalized. He gives patient histories on them with acute liver disease and liver failure from a variety of different diagnoses in the hospitals in New York. And so what he decides to do, because these people all have low hippuric acid output, and he had already found that the average, quote, non-diseased person has a very narrow range of variance in their hippuric acid output based on daily living. But these people had extraordinarily low levels. I mean, 2 standard deviations below other people. And so he then said, well, maybe these are people who are suffering from a functional molecular defect called glycine insufficiency. Now, glycine is the simplest amino acid of all, right? The R group is a hydrogen. And so no one thinks of glycine as being an essential amino acid, at least I don't believe so. But if then, his hypothesis, if I added glycine and elevated exposures to these individuals, I therapeutically administered glycine, could I activate hippuric acid as a surrogate marker for improving liver function? And lo and behold, 70% of those patients on glycine supplementation or glycine therapeutics had resolution of their liver failure. Yeah, 70%. And so this became known as the hippuric acid test for liver function. Now, the reason that I'm saying this is that this led me and my associates, this is when we first started, I left Pauling Institute, gave up my faculty position. We started to how could we do some research? I said, well, let's start studying this. This is kind of interesting. Because we can get sodium benzoate capsules and we can give them to people. It's a GRAS material. I mean, you might not like it, but it is generally regarded as safe. And we'll start measuring hippuric acid in the urine. Simple chemical analysis. And lo and behold, when we did that, we found extraordinary variations among people based upon their health status with regard, and they weren't all liver failure people, just people that had all sorts of generalized symptoms. Then I said, well, hold it, if you can do that, can you measure certain aspects of phase 1 using some kind of a challenge test? Because this is challenge testing, right? You give something and look at the response to it. So the other test that was available, that was GRAS approved, was caffeine clearance. So, and we had no dose. So we said, well, let's get no dose and start giving it to people and measure the rate of excretion of metabolized caffeine or unmetabolized caffeine in their urine. Simple test, things that we could do with our simple equipment. And lo and behold, we started seeing extraordinary variation in caffeine clearance, and it would also couple together with that which we saw with sodium benzoate conversion to hippuric acid. So then I said, well, what other kind of phase 2 things could we examine clinically just to get a beat on this? And I said, well, how about sulfation? So then I went to the literature, started looking at sulfation, and I eventually hit on several articles that I think had been kind of buried in the literature around sulfate to creatinine ratios in the urine as being a surrogate marker for sulfation capability through the sulfation enzymes, PAPs, that add sulfates to things, that the sulfotransphorases, that ultimately leads to one of the pathways, phase 2 detoxification. And that actually turned out to be correlated also with glucuronidation. Wow, now we are, not glucuronidation, glutathione conjugation. So now we start to have this little simple panel, and then I was very fortunate to have met in Atlanta, Georgia, in the middle 70s, a gentleman that was just graduating from his PhD, he and his wife, from Emory, Alexander Brawley, who was setting up a laboratory in Atlanta, later to be called Metametrics. And so I said, so Andy, how about if we set up a collaborative project and we'll start using these tests to see if we can correlate individuals' clinical presentation using a symptom questionnaire, a phenotypic questionnaire, with their ability to do these things, right? Well, that led then to the study that Dr. Minnick reported to you that we published in 1996, the first clinical paper ever been published in Alternative Health and Healing, which was a clinical trial on patients with chronic fatigue-like symptoms on a normal diet versus an augmented diet that contained all these nutrients that would activate those processes associated with phase 1 and phase 2. But what you didn't see probably in Dr. Minnick's presentation was that we actually, and I'm very proud of this when I think back, in 96 reported caffeine clearance, benzoate clearance, sulfation urinary excretion, and we also did lactulose mannitol test to see what their gut integrity was. And what do you think we found in 1996 in that group of over 100 different patients, some in a placebo diet or a control diet and some in the intervention diet? We found that all of those parameters were improved, reduced gut permeability, improved phase 1 and phase 2 clearance, and improved sulfation. Now, with that information, I became an advocate for the concept that I call metabolic detoxification, only now for the last 25 years to sustain extraordinary criticism by the part of many who don't understand this concept at all. They haven't read any of those papers. They haven't done any studies since that work, that ancient work, to see what's gone on in the field as it relates to personalization of concepts that would lead to normalization of the inducible enzyme function. And I want to emphasize, we know there are two types of genes in our bodies, two gene types that code for protein. There are those that are constitutive. Those are called housekeeping. Those genes don't get up and down regulated very much. They kind of just chug along doing their work, housekeeping genes. And then there are these inducible genes that can be up and down regulated based upon the experience of the environmental exposure, right? And many of these cytochrome P450s and phase 2 naphthoquinone reductase, as Dr. Cohen was talking about the other day, these particular genes are regulatable by being exposed to certain kinds of stress factors like glucosinolates, like sulforaphane. because these are agents that the body sees as a foreigner in a way that it then upregulates specific components of this complex process of protection called detoxification. Am I making sense here? So when I put all that together, then in 1991, one of the world's foremost immunologists, clinical immunologists at London College in London at at Bartholomew's, that's right, John Hunter, J.O. Hunter. So you can Google J.O. Hunter. He's highly published, hundreds of articles in clinical immunology. He published an article in The Lancet in 1991 that is a seminal paper. And it was entitled Food Allergy or Intrametabolic Disorder, ? Now, what did he say in that paper? He said a lot of what we're calling food allergy isn't allergy at all from a strict immunologist perspective. It is food response that induces changes in the intrametabolic processing of the materials in that food. So he goes and talks about chocolate and red wine with tyramine. That's not a food allergy. That is a food toxicity related to a component of the food, tyramine, that if you have a certain MAO personality, monoamine oxidase, you may be much more sensitive because you don't clear tyramine from your body. So it's not an allergy. You may present like an allergy, but it's not an allergy. It's a intrametabolic toxicity. This is 1991. So from this construct, what I've just described to you, we started to build out a series of nutritional interventions. And in fact, I just this last week had sent to me from a gentleman that has been coming to my seminars for over 40 years. It's really amazing someone to have that kind of ability to be able to come to 40 years to my seminars, but yeah, and still talk about it. And he sent me, he says, Jeff, I went home after your seminar the other day, and I was cleaning out my office, and I was finding all these old tapes, these celluloid tapes from your series, your seminar series in the late 70s and early 80s, and I found this one I sent you, and I don't have it on slides, but just believe me, it happened to be the color red. I don't know why we chose red as the color of the front of the series, but it was called Food and Inflammation, the Gut-Brain Connection, 1985. So this stuff has been going on in discussion for a long time, and it's been building up more and more support. And I would have to admit that a lot of what we were doing early on was more speculative and hypothetical and kind of trial and error. But over time now, we don't have nearly this kind of reputation of just being wild out cowboys out there without solid scientific information, as is shown by this indoxyl sulfate article. Because you can change indoxyl sulfate in the urine of an individual through dietary intervention, right? And what does indoxyl sulfate do if it's there? It has an effect on macrophages, which induces inflammation and causes a kidney problem. So this is all now becoming real if you need like a lot of basic mechanistic science to understand it. Now, is there still a lot of room for learning? Of course there is. We're still on the front edge of this, but we have crossed the threshold from where it was just a bunch of wild speculation to where we now actually have demonstrable and testable hypotheses that have tools that can actually interrogate these pathways to demonstrate, or these processes to demonstrate their validity. So just to go back to the slides for a second as I'm closing out, when I look at what Barabasi published back in 2000, when was this? 2007, actually. Wow, that's amazing. You probably saw this article in the New England Journal of Medicine when he talked about we're moving from looking at conditions as single points in time to looking at them as part of a network of biology with different layers, from the genomic layer to the metabolic layer to the phenotypic layer, and even into the social networking area. Remember, he talked about, and we've learned that people you hang out with are likely to have same things wrong with you that you do, because you have a social matrix that reinforces and supports certain habits. So all these things are part of this network medicine. And so we're now seeing the emergence of the systems biology of personalized nutrition that is not just speculative. You can actually start examining all these different levels of metabolic flexibility, phenotypic flexibility, as it relates to organ reserve and the functions that we've been really talking about since, since the 80s, of course. And then we talk about this Kira and Karizian study at Harvard Mass General. Most of you are familiar with this work that I think is really important because it lays down a fundamental clinical and I guess you call it randomized study support for the model that genes and lifestyle and diet interface. It's not like it's all genes or it's all diet. There is an interaction between the two. So this was a study you probably know of, over 35,000 people, 3 different studies that were combined, the data from this, where they had genomic information on these individuals. They looked at, by GWAS, at the 50 top genes that were associated with cardiovascular disease risk. And so they categorized those in high, medium, and low risk based on their genotype, monoallele or double allele for these SNPs. And then they had also information on lifestyle, and they categorized their lifestyle based on diet, smoking, exercise, weight, into high, medium, and low risk categories. And what did they find in this study? They found that those that had the highest genetic risk based on these 50 GWAS-identified SNPs, who had the best lifestyle, had a 50% lower incidence of cardiovascular disease progression than those who had the luck of the draw lifestyle and the high genetic risk. So it would be like a pill that you could take people at high risk and lower their incidence into the phenotype by 50%. Now there's another part of this study that a lot of people miss that I think is really an important feature of this, because people who criticize this kind of work will often say, well, there's no clinical marker of an anatomical or pathophysiological indication. You're just talking about relative risk, and there's all sorts of variables. But what they also measured in a subset of this individuals, a number of people had coronary calcium screening, right? Coronary calcium artery screening. And so they asked the question, this is a pathognomonic indicator of disease progression. And so they asked the question, did the genes and the diet have anything to do with coronary calcification? And that's the data that I'm showing you there in this chart on the right, which shows that if you take the highest genetic risk group, that's the ones, you see the bar in the red, and compare it, that's the group, the highest genetic risk that has the highest, or the worst lifestyle. So the red bar on the far right is bad genetic risk, bad lifestyle. Then compare it to, on the left of that, the lower bar, the blue bar, which is the same genetic risk, but the best lifestyle. And what do you see there? The error bars do not overlap, right? There's a statistically significant reduction in coronary artery calcification as a pathognomonic indicator of the disease progression when you have the same genetic risk, but you have a favorable lifestyle. So this then begs the question once again, are there specific things you can do with these individuals that have genetic risk that would even squeeze out better benefit if we were to personalize the therapy? And so I think what we're witnessing is this transition from nutrigenomics to personalizing diets. And this is the American Journal of Clinical Nutrition just a couple of months ago, this article in which they said the best genetic test using single SNPs will never predict a multifactorial outcome of the interaction of multiple genes with lifestyle, diet, and environmental factors. However, the prediction of disease risk is much less important than the understanding of genetic uniqueness of that individual, how they process specific nutrients, and how that impacts their function. That's a quote from this article. So we've been misusing genetic information, I believe, in the way that it can be applied to personalized assessment and looking at metabolic groups of functions versus looking at disease risk. I think that's where this is heading, which takes us into a thing like this Framingham study that was just, again, recently published, looking at individuals that were on healthy diets versus unhealthy diets and looking at their metabolomic profile. In other words, what happened to their metabolites versus an unhealthy diet group? Can you actually predict certain changes and shifts of the fabric of metabolism, which is the reflection of what's going on at the gene expression level, obviously. So what they say is healthier diet was associated with genes involved in metabolic function. Further work is needed to replicate the findings. However, this does suggest that healthy diet has a direct effect on gene expressions into healthy function. So we're starting to get the tools to interrogate that. So let's go back to Kira and Kazizran at Mass General at Harvard again. These are the same authors that I showed you earlier with the cardiovascular disease risk. So what is this study? So they wanted to know something about obesity genes. And this plays very beautifully with what Dr. Knight was telling us earlier, that you really get very poor connection between genomics of the host and the propensity towards obesity. It's much closer if you do the genetics of the microbiome. So what did they have to do in this study in order to get a clinical correlation between genetic profiling, this is eukaryotic genetic profiling, and obesity risk? They can only do it if they didn't actually focus on the GWAS identified genes for obesity that had no relevancy really when it was translated into the individual in terms of clinical risk to their own obesity. They only got a clinical correlation when they did an analysis of 1 million genes. 1 million genes. Did you hear what I just said? That goes beyond polygenetic to omnogenetic. Now, why is that? Because obesity is a phenotypic manifestation of an extraordinary array of different metabolic genomic alterations, which interfaces with the microbiome. And so you have to have a much broader net to throw over all of this to understand it, rather than saying, oh, you've got obesity genes. When I heard James Watson a number of years ago, you might remember this, there was a big press release on this that we have discovered the gene for autism. I could not believe that James Watson, the father of the gene, would say something like that. How in the world did he say that when we know that that was stupid from the very beginning of its statement? And it was as stupid, quite honestly, as saying that the dark matter in the genome is junk DNA. That was stupid too, wasn't it? And I'm very proud to say that over the 25 years I've been talking about genomics, I have said, don't consider that dark matter to be junk. You don't have, only 3% of our genome codes for protein. You know that, right? All that other stuff is not in there just to occupy space. It's the executive function that regulates through small inducible RNAs and all sorts of regulatory elements. It actually is the executive center that controls how genes are expressed multiples at a time. I've often asked the individual, I said, how many permutations and combinations can you think of 25,000 genes expressed in multiple combinations at a time? And the answer is virtually infinite, right? It's not like one gene at a time. You have 20. So that probably explains why the Pinot grape has 40% more genes than we do. 40% more coding genes in the Pinot grape. That's why you love Pinot so much, right? It's very rich in its phytochemicals. It gives rise to all these flavors. So you might say, well, the Pinot grape is a lot more sophisticated than we are. But it's the transcription control of gene expression that is included in the dark matter of the genome that really gives us our diversity and our uniqueness in the species of animals and plants. So Rob said it all. He said it beautifully. If you were to say to me, what's the takeaway from the Knight extraordinary discoveries that will ultimately, I believe, win him a Nobel Prize and his group? It's now we're starting to see that translation into some operational execution. He had mentioned that, I believe in his disclosures, that he is a consultant to Day 2. Day 2 is a company that's been formed from the group at Weizmann in Israel that published this landmark paper from their work in 2015 in Cell around personalizing nutrition based upon an algorithm developed from the gut microbiome that predicts the glycemic response to foods that is almost as accurate, and in some cases more, than continuous glucose monitoring. This is by measuring the gut microbiome and passing it through this algorithm, really machine learning. So there's many papers on this. This is assessment of personalized approach to predicting postprandial glycemic response among food in individuals without diabetes. This appeared recently in JAMA network, showing that you could do predictions based upon this algorithm that is derived from gut microbiome shotgun sequencing. Not single genes, but really looking at shotgun sequencing of the gut microbiome. And this is another one of their articles, because people said, well, gee whiz, that work was done in Israel. So maybe Israeli people are different than people in the United States. So how can you extrapolate? So this is a companion study done on Midwestern United States people in conjunction with Mayo, showing they got the same results using this kind of an algorithm. So we're witnessing now the emergence of the ability to harness this technology and translate it into clinical prediction. So yes, this phytochemicals that we've discussed and heard so much about over the course of the last 3 days are extraordinarily important. we have about between 100 and 150 known nutrients. Some of those we call essential, some are non-essential. But if you actually look at what's called the dark matter of nutrition, as you've heard, it's a range of all these phytochemicals that Deanna started us off thinking about in these colored compounds, of which there are 28,771 compounds that have been identified from foods that are not essential nutrients that modulate biological response. This is a treasure. This makes the lexicon of pharmaceuticals pale in comparison. And we're just learning for the first time how these affect gene expression, epigenetics, metabolic regulatory processes. So let me just close. This is a topic that's been close to my heart for some time. I know that many of you have heard about this, but that's the chemosensory receptors that we labeled as bitter, sweet, salty, and umami receptors. We think of these as taste receptors. I'm going to tell you, throw that term away. It's very limiting. The reason that we call them taste receptors is that were first identified in the tip of the tongue, and neurophysiologists studied how these receptors could activate certain neurological pathways that were associated with the perception of those characteristics: sweet, salt, bitter, sour, and umami. But now, thanks to the work that Wolfgang Meyerhoff and his group has done in Germany, who is probably the world's recognized leader now in bitter receptors, we start to recognize that these are not just localized on the tip of the tongue, that these receptors are found throughout the whole of the body. So, in fact, and this is, when I say this, I know this gets a lot of people's attention, they are found on the ovaries of women, so ovaries are tasting. Right? So now you start to say, well, hold it just a minute. Why do we have bitter taste receptors on L cells of the distal ileum, beta cells of the endocrine pancreas, hepatocytes, macrophages, adipocytes, I think I said adipocytes, and on reproductive tissues? Why are they there? They're there not to taste. They're there as chemosensory receptors to pick up information signals from the environment and translate it into function at that multiple organ system of crosstalk. And that crosstalk is what you eat, how the gut responds to what you eat, how it then influences the enteroendocrine system with the production of GLP-1 and GIP and and cholecystokinin and all these various enteroendocrine hormones that are produced where? Produced by specific cells, many of them in the gastrointestinal mucosa. GLP-1 is produced by the L cells by response to the bitter receptor 38. There are 50 different mammalian bitter receptors. Some of them each one does a different thing. So these are chemosensory receptors that regulate crosstalk among multiple tissues associated with the neuroendocrine immune system. Now think of eating. I mean, why did these things all evolve to have this complex interplay? Was it just no better place to put them, just throw them in there and we'll see what happens? No, they're in there for specific signaling crosstalk to interrelate things like bacterial infection and PAMPs and DAMPs and how they get responded to by different tissues. So maybe they're in reproductive tissues because when you're under high stress of an infection, reproduction is not your first order. You ought to be sending energy into other places, so you regulate function. Why are they in the adipocyte? Why do you have angry fat and friendly fat? Because you're activating toll-like receptor 4 on the surface of a adipocyte that crosstalks to a crown cell macrophage because the embedded activated immune system into your subcutaneous, into your mental fat activates that processes through intracellular signal transduction, so you're getting a crosstalk that also speaks to the brain's immune system, which is called the microglia, i.e. gut, brain, liver, immune system, all interconnected. And yet we're still thinking about these as individual chapters in a textbook and shutting at the end of each chapter and taking a test, thinking we know anything. So I think that this construct, this is a paper that just appeared actually in American Journal of Clinical Nutrition that really bears on my point. They looked at the association between taste perception and adiposity in overweight, obese subjects, and they found that there was a specific bitter taste receptor polymorphism that had a more strong association with obesity, which happens to be the bitter taste receptor that we know is activated in the gut L cells to produce GLP-1, which is an insulin stimulating hormone, and has an effect on anti-inflammation, right? So these things that we are learning, and I'll just say I'm very proud of this publication. I'm one of the co-authors on this recent publication. Intestinal bitter taste receptor activation alters hormone secretion and imparts metabolic benefits. Here we showed in an animal model of polycystic ovary syndrome that by modulating the way these bitter receptors are activated, we could actually change androgen synthesis at the theca cell in the ovary. Meaning we could treat hyperandrogenism with modulation of bitter receptors in the ovary. And how did we do that? We did that because we used isohumulones. And isohumulones happen to be one of those multiple thousands of compounds I talked about that are in plants. Isohumulones are in the bitter component of hops, Humulus lupulus. And so, all of my associates here recently were involved with this publication on the pleomorphic effect of these bitter substances that modulate their activity through this bitter receptor signaling system. So I'm going to pass through this. So that then will finish up with what you already heard from Dr. Ben-Akhtar, which is when you start then looking at this complex interplay we have with our environment, both exotoxins and endotoxins and the total load, fortunately we have this adaptive system that's upregulatable called the detoxification system. And it's barriers of defense, it's these different enzyme systems that regulate function and help to get stuff exited out of the body before it does a lot of mischief. And these processes are in part related to how we eat, but how we think, act, and move, and where we live. All of these are variables that influence these things. So in this case, as Dr. Ben-Akhtar showed, when a group of individuals was given this broccoli sprout beverage, and by the way, this included investigators from Johns Hopkins. You recall the group that was originally doing their broccoli sprout work. So they're represented in this study, Jed Fahey, and et al. And what they showed was increased benzene excretion with dose response exposure to a beverage of broccoli sprouts, right? So it was like a pharmacological study. Increased exposure, increased excretion. And so that lastly then ties to things like this. It starts to make us understand maybe these associations. In this case, broccoli soup and its effect on prostate cancer gene expression. So here is where you take human prostate cancer cells from biopsy and you expose them to a broccoli concentrate and you look at the changes in gene expression. Then you give the broccoli soup to a human who has prostate cancer and you see if it will play out in the whole organism, what you saw in vitro, and lo and behold, they map together. They map together. So these are extraordinary thresholds that we're crossing, never to go back again. And when I say it's all changing, it's all changing. It's all changing. This isn't a fad. It's not like here today, gone tomorrow. It's not like, oh, that was an interesting thing we learned yesterday, but tomorrow we're going to back where we were. This is a one-way street of discovery that is making so much of what we learned about determinism wrong. We now recognize the plasticity of our genetic expressions. We now recognize from telomere-link studies and from various types of work that's now done on mutations within immune cells like 1011 translocase and DMT3, which are epigenetic enzymes that are modulated in our immune system to cause chronic inflammation that are a result of the exposure to things in our environment that mutate these genes and form clonal populations of white cells that, as we get older, we get more inflamed. Now we're starting to understand the mechanism. And by the way, if you really want to drill deep with us on, that's the focus of my 2020 October meeting, is going to be on really looking at how these things that we've now learned about clonal hematopoiesis of indeterminate potential really happen and how diet influences them. So this is a threshold that we're crossing that we're never going to go back over again. So in closure, the genes that have been identified, I'm quoting now from this article, the genes that have been identified as being very important in this nutrigenomic gene-environment interplay play roles in lipolysis, that's the breakdown of fats, thermogenesis, glucose tolerance, and detoxification. These are quotes, by the way, from this AJCN article. One of the most significant genes they talk about is this ARDC3, which is known to play a role in regulating cell surface expression of adrenergic receptors and other G protein coupled receptors. And I want to just take a moment to say what that means to me. These are GPCRs. There are hundreds of different GPCRs that sit on the surface of every cell in our body that are the antennae that pick up information to transduce signals to our genes to tell them how to regulate their function. And those are, many of those G protein coupled receptors are nutrient modified in their function. Let me give you one example that now is well understood, omega-3 fatty acids. We're now starting to see that the mechanism of action of omega-3s is through a specific members of the G protein coupled receptor family that transduces across the membrane. It's a 7 transmembrane protein. The interside of that membrane then is involved with activation of kinase signaling processes that regulates gene expression through these transduction pathways. You've heard things like mTOR, and there's all sorts of things going on now about how nutrients modulate these various things. So lastly, they say, and I absolutely agree, these results can leverage genetic expression analyses to provide early biological footprints, I would call it thumbprints, but okay, footprints, of an unhealthy diet environment for the individual. This is the threshold of personalized nutrition. We're not now to the end point, but we're a journey that has crossed the threshold never to go back. And anyone that brings up and counterpoint to this, that has not read this literature, has not bathed themselves in this information, that thinks that they are right in reading a textbook from 10 years ago, is an artifact going to the happy hunting ground of extinction. Thank you very much.